Hello, hello. Hi, everyone, and a warm welcome to today's research seminar. So as the convener of the RITMO seminar series, I'm very pleased to introduce today's speaker, Professor Caroline Palmer, who's the Canadian Research Chair in Cognitive Neuroscience of Performance and Professor in the Department of Psychology at McGill University. Her research in cognitive science addresses the behavioral and neural foundations that make it possible for people to produce auditory sequences such as playing an instrument or speaking. Palmer has developed and empirically tested computational models of how people perceive and produce auditory sequences and how they coordinate their actions with others. And I'm sure we'll hear much more about those aspects today. The title of the paper, and Caroline will speak for about an hour, and then there'll be space for discussion and comments and questions afterwards. And the title of the paper is Interpersonal Synchrony in Sound, Brain, and Body. So please join me in extending a warm welcome to Caroline Palmer.
Thank you very much. I am used to lecturing in big halls, and I have a big voice. So don't be shy. Ask questions as we go if you wish. I'm going to start out with some thank yous because one often gets to the end and runs out of steam for the acknowledgments. The work you hear about today is a function of a really talented team of collaborators, mainly at McGill, but also at Oldenburg, Illinois in the US, Buffalo in New York, and Aarhus. I also want to thank our hosts, or my hosts, Anne and Alexander, because it takes a lot of work to run a center. And RITMO is booming with energy, and this is a wonderful place for me to spend part of my sabbatical. And I know how much energy it takes because I'm in my 10th year of running a training grant, which is a similar size. So kudos to Anne and Alexander. Okay, so Canada is a lot like Norway, and it has made me feel comfortable to know that you have multiple climates. You have an advantage in some ways. You have two winters, I am told, the dark winter and the light winter, the main difference being the portion that is snow-filled so that the light reflects off of the snow. In Canada, we too have two primary seasons. Oh, by the way, in Bergen, not a cloud in the sky. I'm happy to say. So I hope you experience that as well. This is my postdoc, Valentine, who spent the past month here and had a fantastic time. So in Canada, we have two seasons, winter and construction. And the dreaded Rue Barré sign comes out, and you cannot get to point A from point B. If you look at the map of the island of Montreal and you go to the highway division, that's your average summer day. And each yellow sign means you can't get there from here. So I don't know if I'm going to see construction season in Oslo, but if you come to Montreal, come before the beginning of the construction season. So today I'll talk about coordination of movement with sound. We, as this group especially knows, musical coordination requires very fine temporal adaptation to create synchrony in sound with partners in ensembles. But you can see this emerge in humans without instruction as early as preschool years. And you can see it emerge without intention, such as audience members of this famous example of clapping that tend to become synchronous over time. This coordination of movement and sound arises, in our view, from self-organizing principles of coupled oscillation. So what do I mean by coupled oscillations? So we can look at oscillations in the human brain at the intercellular level. Excitatory neurons and inhibitory neurons each have inputs and outputs, but importantly, there's a feedback loop that enables them to couple with each other. At the population level, we can look at local networks of brain cells in terms of the excitatory and inhibitory patterns of activation that, when combined, generate a neural oscillation at the population level. one of the main principles of neural oscillations is the intrinsic or natural frequency, sometimes called the resonant frequency, because that is the stimulus frequency to which the neural oscillation will respond. So oscillations increase their response to or couple with stimulus frequencies that are close to their natural frequency. And we know from important work in neuroscience that premotor cortex and auditory cortex show coupled regions in individuals with musical experience or performing a musical task. So what kind of evidence is there for these biological oscillations and natural frequencies? If you look at animal species such as cockatoos, such as Snowball, who bobs his head to a certain rate of music, or Ronan, the sea lion, who was trained also to bob his head to a certain rate of music, you'll see biases towards a natural frequency at which there is the greatest stability of the oscillation and an increased response to stimulus frequencies. So spontaneous rhythmic movement to periodic sound is seen in many animal species. A good example is Bengalese finches. So they are songbirds who are tutored by the father. And the father's song tempo in syllables per second is highly correlated with the offspring's song tempo. Now, Metz and Brainerd were able to do careful experiments in which they can raise the birds in an environment with just one song tempo. When they did that, they found that the bird's eventual song tempo still matched that of the father, which Metz and Brainerd took to indicate the genetic contributions to this spontaneous frequency. Notice the big range of individual differences among the birds, yet they are consistent in producing that bird's instantaneous, spontaneous frequency. So human rhythmic behavior is much more flexible. We are capable of a much wider range of rates and more adaptive. We are able to adapt to people with other rates. Yet there are still motor signatures, which von Holst proposed as the intrinsic dynamics that make my patterns of movements specific. So I'm going to, because I know that this is RITMO and talks often try to tie to real world examples, for me, that is Jean-Baptiste. Jean-Baptiste is a performing musician, composer, conductor, record producer, dancer. He is a polymath. He has a specific pattern of movements that are intrinsic to his frame. And I'll play you a That's great. How many of you are familiar with this song? Only some. Okay. Well, Jean-Baptiste is highly entertaining, and I encourage you to look at his videos, listen to his records. In this song, he talks about the movements that he makes and the feeling that he has as he's dancing. So I highlighted the words especially because it's the way that you walk, you can't contain it. Is it the shoes jumped up, kangaroo? These are discussions about what I would consider intrinsic motor signatures, intrinsic dynamics of the body. And if you watch one of his videos, you will see that he makes very specific moves that I'm not capable of making. You can see them. So this is a real world example of what I'm going to talk about today in auditory motor integration. So the outline of my talk is I'm going to talk about spontaneous movement rates of musicians that reflect basic oscillator properties. We'll talk about musicians and non-musicians in showing these spontaneous rates. We'll talk about the temporal stability that they show at those rates and tempo drift or a bias towards those rates. I'll compare non-musicians with musicians in a novel task that they're both able to do. And then we'll turn to interpersonal musical synchrony, which is our ultimate goal. Can we explain why some partners are better matched to other partners based on those spontaneous rates. And then I'll talk about a neural marker that corresponds to those spontaneous rates. Okay, so how do we measure these? This was a serendipitous finding of Janine Lair. We found that certain performers were always veering in the direction of a specific tempo, but they weren't veering in the same direction. So we tried to measure this more systematically by asking each person to perform at a spontaneous rate with no cues, whatever rate was best for them. We're considering those spontaneous rates a proxy for the natural frequency of an oscillator. So the task might be to produce a simple sequence, a short melody at a comfortable rate from memory. It's always from memory because we're trying to remove the possible influences of musical notation on the rate at which they play. So I'll play you an example of an unfamiliar melody. It's actually a canon by Thomas Tallis, presented to a group of performing pianists, each of which chose their rate. We took multiple recordings. The mean tone duration, or inter-onset interval, IOI, is on the y-axis, and the performers are ordered from fastest to slowest. So here's an example of one of the faster ones. And one of the slower ones. Now, those of you who are musicians know that one question that arises could be, are they interpreting it at a different level in a hierarchy of meter? For instance, are they emphasizing the quarter note versus the half note? If that were true, you would expect to see a multi or bimodal distribution. But instead, we see this fairly continuous, smooth distribution across the musicians. So we don't think this is simply a matter of interpreting different tempi or different metrical levels as more important. Now, the best way to test this is to ask them to play different pieces, and that's what we did. And so we compare their rate of performing one piece with their rate of performing another piece. Again, a novel piece that they have some time to practice, memorize, and then we take the music away. You can see that the correlation across pieces is quite high, but the difference is on an order of 2-to-1 or 3-to-1 difference in tempo from the fastest to the slowest performer. So we've shown that these spontaneous rates vary across individuals and they're consistent with individuals, but is it specific to something about piano performance or perhaps finger movements? So we compare it in singers. In this case, it was with a familiar melody, and you can see that the distribution shows a similar pattern from the fastest to the slowest, and in a smaller sample of violinists. Now, singing, playing piano, and playing violin are quite different movements. So we think that this is something that is specific to the individual, not necessarily to the instrument. But we would like to know if this is specific to music training. So my PhD student, Becca Shurik, developed a task of allowing individuals to press a force sensor and which generated the next tone in a melody. So this was a way that musicians and non-musicians could control the rate of familiar melodies. So this task is only possible with familiar melodies, but it's a novel interface for both the musicians and the non-musicians. So in this case, we don't have to limit our groups to pianists, singers, or violinists. We can compare musicians of all instruments as well as non-musicians. And she called this task Piano Hero, to take off on Guitar Hero, if any of you are familiar with that. So when we asked the musicians and non-musicians to do one finger tapping where they controlled the rate of the melody, we see the same kind of distribution. And there is some evidence, but it's not strong, that the non-musicians tend to go a little faster than the musicians. This has been shown in other studies, but in this case, it's not significantly faster. Okay. So if we compare these spontaneous rates across pieces, they look very consistent. If we compare it across hands, where we ask pianists to play some melodies with the right hand and a different set of melodies with the left hand, we see the same consistent differences across the individuals in the rate at which they produce, the spontaneous rate for these simple melodies. We brought some of the musicians back a year later, up to a year later. And this graph shows that you start seeing some dispersion, but it's still significant, the relationship between their first recording and their later recording. And this is the work of Anna Zam, who I understand you may be hearing about in a few weeks at RITMO. So these spontaneous rates look pretty consistent. So where might they come from? Well, one view, very popular now, is that temporal coordination should be easiest at the rate that requires the least energy expenditure. So this relationship between high and low effort and high and low levels of performance should show an inverse relationship. And people have applied this to tasks such as walking. So each individual has a certain leg length and a certain gait pattern. And if you ask them to walk at an optimal rate, and then you ask them to walk faster or slower than that rate, you will see that the oxygen consumption goes up, whether it's faster or slower. And if you think back to the last time you walked with someone who walked much slower than you, perhaps a young child, it's tough to walk at that speed if you are used to walking at a faster rate. So it's difficult to walk both faster and slower than the optimal rate for individuals. And we took this view to indicate that we should see the greatest stability in performance at these spontaneous rates. So we're going to make the prediction that performers should show less temporal variability when they're performing a melody at that person's spontaneous rate. So in this experiment, we asked pianists to perform a melody first at a natural pace, and then we would either ask them to play it faster, faster again, slower, or even slower. And we balanced the order in which they did these conditions. So here's the mean rate, or mean IOI, intertone interval, at which they played, and it shows that they followed the instructions. They were able to play slower, even slower, faster, even faster than their natural rates. But the interesting thing is the temporal variability. What happens to the timing of those tone onsets when they have to play at a rate that's faster or slower than their optimal rate? You see the same kind of U-shaped curve that you see in the upper right from the oxygen consumption during individual walkers trying to walk slower or faster. So this finding suggests that indeed musicians' temporal stability is greatest at their spontaneous rate. What about what happens within a performance? Because these nonlinear dynamics that are represented in natural frequencies of oscillations should have some prediction for change over the course of the performance. So we look at the serial position in the melody for evidence that they should show some drift back towards their optimal rate. So if they're playing at too fast a rate, they should show drift slower. And if they're playing at too slow a rate, they should show a drift faster. So we're looking at the slope of the tone durations across the serial positions in the melody. And that's what's shown on the y-axis here. So they slowed down. They have a positive slope when the cued rate was faster than their spontaneous rate, and they sped up when the cued rate was slower than their spontaneous rate. Okay, so that's consistent with the idea that it's this ongoing oscillation that is driving performance, and we see biases towards individual spontaneous rates. We've replicated this with violinists and with tappers in the Piano Hero design. So it's not specific to a musical instrument or to a limb. Okay. So maybe these spontaneous rate differences could be specific to motor performance. Are they really specific to auditory motor integration? There are two highly related tasks that are used a lot in the literature. One is called the spontaneous motor tempo, and individuals are asked to tap regularly without any sound to figure out what their spontaneous motor response rate is. And we have this spontaneous production rate task, like Piano Hero, where there is sound involved. So we asked whether or not these two generate similar values in individuals. Are they correlated? And first, I'm going to show you the difference in their values. So here, the spontaneous rates are ordered from smallest to largest, or fastest on the left to slowest on the right for the musicians and non-musicians. And in the top, you have their corresponding spontaneous motor tempos. So the participants in the top graph are also ordered by the spontaneous production rate in the bottom graph, the similar orderings. And you can see that the same relationship doesn't hold. There's no correlation between the spontaneous production rate and the spontaneous motor tempo. So this suggests that the spontaneous production rates we are measuring are specific to auditory motor integration, not just to motor performance. Okay, so the prediction from the coupled oscillator approach is that there should be better interpersonal synchrony between individuals who have similar natural frequencies. If we look at pairs of individuals in the graph in the upper right, those who are close to each other should find it easier to synchronize than individuals who are farther apart. And so in this study with Pauline Tranchant, a postdoc in the lab, she randomly assigned individuals, musicians or non-musicians, to pairs. A non-musician was always paired with a non-musician, and a musician was always paired with a musician. And they performed a familiar melody, first in the solo performance, and then in the joint performance. At the point of the duet performance, they're both playing on the Piano Hero task because we need something that non-musicians can do also for comparison. They're first cued at partner one's spontaneous rate from the solo performance, and then they're cued at partner two's spontaneous rate from the solo performance. So we've measured the two partners individually in their solo performance, and we use those spontaneous rates to cue the joint performance. Because it could be that one is only good at playing at one's own rate, or it might be that musicians differ in that ability versus non-musicians, as you might expect if musicians are more temporally adaptive. So I'm going to play you a pair. I'm going to play you two of the duet performances, and I'll ask you to think about which is a musician pair and which is a non-musician pair. That's the cue. Okay, now pair two. Okay, I see some smiles, so I think you know the answer. How many thought that the first pair were the non-musicians? How many people thought that the second pair were the non-musicians? Okay, it's overwhelmingly in favor of the second pair, and yes, they were indeed the non-musicians. So this follows from the fact that the coupling should be greatest or easiest if you are playing with someone who shares a natural frequency, where the difference in the natural frequencies is small. Then coupling should be easiest and synchrony should be highest. So I'm going to show you a graph that shows on the x-axis the difference in the partner's spontaneous rates in the solo task, and on the y-axis the mean asynchrony in the duet task. Because remember, we're trying to predict the amount of asynchrony from the difference in the natural frequencies. And so one important pattern to show is that they're both positive slope lines. So there's a directionality to both the asynchrony and the difference in the spontaneous rates. The second pattern to note is that the non-musician's slope is significantly steeper than that of the musicians. If the musicians were perfectly adaptable, they should show a flat line, right? So the slope tells you something about the adaptation, temporal adaptation abilities of the participants in the task. Now, in the second part of the experiment, where we play the cues at partner 2 solo rate, you see the same pattern, but now the non-musicians and the musicians start looking like they have similar slopes because they're getting better at it as they go on. Importantly, the amount of duet asynchrony is not predicted by an individual partner's rate. It's predicted by the difference in the two rates, even though they're only playing at one rate at that time. So that's in support of the idea that coupled oscillations arise in situations where individuals share these natural frequencies. Okay, so just to summarize what we've gone through so far. Partners with similar natural frequencies were more synchronous. This is true for musicians and non-musicians. Both partners' spontaneous rates predict the duet synchrony. It's not just attributable to one partner. And we have replicated this pattern with piano duets and with vocal duets, so it's not specific to the limb mechanisms involved. But I haven't addressed whether or not there's a causal relationship, because these are all correlations. So we want to show a causal relationship between the spontaneous rates and the interpersonal synchrony in the joint performance. So in this experiment, we took pianists and we first measured their spontaneous rates. And then we created pairs, matched pairs, who had a difference of a mean of 1% difference in their spontaneous rates, and mismatched pairs who had a mean of 38% difference in their spontaneous rates. What's important to note is that the y-axis, the absolute rate, is similar. The range of absolute rates on the y-axis is similar for both the matched pairs and the mismatched pairs. So they perform in the solo context. They are paired deliberately with a partner who matches them or mismatches them in their spontaneous rates, and we bring them in to perform together. So this is a graph of the amount of absolute asynchrony in the duets for the matched pairs in the black bars and the mismatched pairs in the white bars. You can see that it's consistent across the melody repetition, so that it's not something that they can quickly adapt to. And in fact, the matched pairs are more synchronous than the mismatched pairs, although both groups are doing pretty well. This is a small amount of asynchrony. It's consistent across repetitions, and there were no differences due to the limb used. So this was a piano performance task in which half of the time they were playing a melody with the right hand, and the other half of the time they played different melodies with the left hand. So the limb did not affect the amount of asynchrony. It was the natural frequencies that differed. Okay, so that suggests there is a causal relationship between the amount of asynchrony in the duet performance and the individual spontaneous rates in the solo performance. So we think these spontaneous movement rates reflect basic oscillator properties. They are consistent. They show a bias over time. If you're playing at a different rate, there's greatest temporal stability at that rate. Non-musicians tend to be more fixed than musicians, although that changes with practice. And you can predict interpersonal musical synchrony from the spontaneous rates. So now I'm going to turn to neural markers of the interpersonal musical synchrony. There are several alternative explanations for spontaneous rate differences. One is to think about peripherally driven timing, the idea that there are limb-specific rhythm generators, and they are associated with anatomical and neuromuscular mechanisms or asymmetries that differ across individuals. But we didn't see too much evidence to support this because there were no arm or hand differences in the musicians' natural frequencies. And we saw similar influences across musicians and non-musicians, suggesting it's not simply skill level that accounts for it. Another approach is centrally driven timing, the idea that there is a central pattern generator that emerges from networks of oscillators that couple. And this is not peripheral, this is central, so we're talking in the brain. So we're going to actually test this approach by looking at EEG activity in auditory motor networks as musicians perform. That's what I'll talk about next. I will be talking about the other elephant in the room, predictive coding, but not today. And if you're interested, let me know and I'll send you a Zoom link. Okay, so this is work with Anazam, and we were interested in using techniques that have been used in other EEG studies that show increased power present in the brain at auditory stimulus frequencies. And this is work by Sylvie Nazarad and colleagues, and she's shown it while individuals listen to or tap with auditory rhythms. So you can do a fast Fourier transform and look at the power present at each frequency that is present in the stimulus. And you will see a bump, an increase in the power in the EEG signal present at the beat frequency of the stimulus. So we use this type of technique to predict that the power spectral density, that's the value here that we're measuring, should increase at the natural frequency of each performer. So in this experiment with musicians and with wireless EEG, which I understand we're going to see a lot more of in a few weeks here. In solo trials, performers play a familiar melody. Then they're brought together with a duet partner, and they take turns as the leader in this design, where the leader sets the pace. Usually the leader sets a pace similar to what they played in the solo trials. I'll show you that in a minute. And we measure 24-channel wireless EEG. Okay. So the first thing one would want to make sure of is that there are auditory and motor evoked potentials that correspond to what one would expect in this right-handed only melody case. So I'm showing you an example of the motor potential on average peaks around 10 milliseconds prior to the tone onset, which I've got labeled here as key press at time 0, and an auditory potential, which peaked on average around 204 milliseconds after the key press. And you can see the motor potential in blue for the topographic map looking down on the head, where the nose is at the top, and the auditory potential, central, slightly left lateralized for this right-handed performance. So how do we measure the PSD, the power spectral density markers in the solo performance? Well, now instead of showing you tone duration on the y-axis, I'm showing you hertz, because we're going to compare the frequencies in hertz or cycles per second. I'm showing you the tones, the number of tones per second on the y-axis here for all of the pianists in the study. And I'm highlighting this time the slow end is at the left and the fast end is at the right. So I'm highlighting 4 examples to show you what it looks like as you walk through the analysis. So this is power on the y-axis for just the 4 individuals who are highlighted in the top. And you can see the performance rate differs now, again, in hertz or tones per second. And you see peaks that correspond to the order in which those solo performances were played. Now we take those and we align them so they're all centered at their natural frequency. So the 0 point here is the frequency bin that corresponds to the performance rate. That is the behavioral performance rate. And you can see that there's an energy peak that corresponds to the behavioral performance rate. The green line is the grand average, and the red line is the mean at the region of interest, ROI, which corresponds to the auditory ROI in this topographic map. Okay, so now what we want to know is, are they specific to the spontaneous rates of the performance? The only way to measure that is to look at the same frequencies in other people who did not play at that spontaneous rate. So that's what this graph shows. For each of the pianists, it shows the EEG power present at that pianist's spontaneous rate, the red bar, and the EEG power present at the same rate in the other pianists who did not play at that rate. That's the blue bar. And so you can see that the power is performer specific. Okay? So these spectral peaks coincide with spontaneous rates, and they are performer specific. Now we want to go look at the duet performances. And we're going to do the same analysis, look for the power spectral density markers in the duet performances. Just to remind you, they took turns being leader, and the leader decided the tempo for that duet performance. So the mean asynchrony across the pairs was pretty low. So they're doing pretty well, but this is the mean. So you will see a range of values. So we do the same analysis, and we look for the power spectral density peak at the performance frequency, marked here by 0, and it shows the same auditory ROI, region of interest. So they're showing the same spectral peak at the duet performance rate. Now, we want to draw a correspondence between the behavior, the amount of asynchrony, and the amount of power spectral density present. So we correlate those two, and we find a significant negative correlation. So the asynchrony grows off to the right on the x-axis as the power present at the duet frequency shrinks. In other words, if a lot of power is present at that frequency, it corresponds to good synchronization of the duet. What is the effect of the individual? Because remember, the partner 1 was the leader who chose the rate in the first set of performances, and then partner 2 took a turn. So we look at the leader's spectral power at the duet frequency, and we look at the follower's spectral power, where the follower is the other individual who has to play at the rate that the leader sets. And you can see there's a strong relationship, negative, between the leader's power and the amount of asynchrony. And when that changes, so the partner becomes the leader, that same relationship holds, even though now they're playing at different rates. So the follower's spectral power does not contribute to the duet asynchrony significantly. The follower has to adjust their rate more than the leader. And this PSD marker, power spectral density, is performer specific, not just performance specific. So that's the important punchline here. So how do individuals coordinate their auditory motor behaviors? We think these natural frequencies are stable, efficient coordination modes. They are auditory motor integration that arises via coupling of these natural frequencies. And we think this is a neural correlate of the synchrony that arises from the spontaneous rates. So the implications here are that there are partner-specific differences in group tasks. And the amount of flexibility or adaptability can improve with practice and training. So there are pedagogical implications for individuals who want to learn to practice at different movement rates. I want to take a minute and talk about the possible origins of these individual differences in natural frequencies. We're not able to do nice, elegant work like Metz and Brainerd, where we raise babies to hear only one frequency, right? But Paul Frez was the first to suggest that the sucking central pattern generator is one of the earliest rhythms that humans produce. These are spontaneous rhythmic mouthing movements that are critical to survival. If a baby cannot suck, it is not going to survive well. Now, it's not just present during eating or drinking or oral ingestion. It is present throughout the first 5 years of most children's lives. It's something called non-nutritive sucking when it doesn't involve nutrition, NNS. So NNS generates sound, both through bone and air conduction. It's almost impossible to suck without hearing sound. Try it sometime. So in utero, NNS, non-nutritive sucking, starts as early as 13 weeks into gestation. And it's stable by 34 weeks. We're talking on a 40-week clock. The individual differences in non-nutritive sucking, which is measured by a pacifier in the bottom right here, that measures the force with which the infant is sucking. And if you've put your finger in an infant's mouth, you know that they are very good at sucking. So these individual differences range from 1 hertz to 2.6 hertz. That mean of 2 hertz corresponds to the 2 hertz mean across the range of pianists, violinists, singers, and tappers that I showed you earlier in the talk. So there is a consistency in this non-nutritive sucking frequency. It's been measured from fetus to 4-week-olds, and it's been measured from 3-month-olds to 12-month-olds to show consistency. There are many benefits of rhythmic NNS in infancy and early preschool years. An NICU, neonatal intensive care unit, that's where most preemies who are at risk end up in a hospital, in an NICU. And the majority of these studies are run in NICUs. If they get their heels pricked all the time to draw blood, and they need to be given something to calm them down. Their sensitivity to pain is decreased if you can induce NNS sucking in them. NNS is used to self-pacify. These are basically studies, nursing studies, that show that one can use NNS to self-pacify a child up to 4 years old. There is a rhythmicity in the NNS between 42 and 50 weeks of gestational age that's predictive of later cognitive motor performance outcomes at age 5. This is a long-term study. This is a true longitudinal study. Rhythmic sucking is really basic to survival, but it has more implications than simply nutrition. So we think that the sucking CPG may be an antecedent of these spontaneous rates because it survives so long into childhood and seems to match the frequency range and yields a link between the motor system and the auditory system. So future work that we're doing involves parent-child spontaneous rate because of the possible genetic influences of the individual differences in sucking CPGs. We're also looking at the influence of these individual differences on special populations, such as rhythm perception in individuals with ADHD, attention deficit and hyperactivity disorder. And finally, we're putting together computational models of group synchrony. And one of my heroes, John Baptiste, he represents the fact that the rhythms found in music offer a wide range of frequencies for coupling movement with sound. And so we try to take this musical dynamics, nonlinear dynamics, and social dynamics perspectives to draw up new models. So before I end, I just want to say, as the director of a training grant, I must always advertise. So our training opportunities are open to citizens of all countries, including postdoc 2-year scholarships and a summer school in nonlinear dynamics, which I think I've spoken with some of you about. Talk with me afterwards if you're interested. And thank you again to Anne and Alexander for having me here. Thank you.
Thank you so much, Caroline. Yep, it's working. We now have time for questions and comments from the floor. I'm going to jump in with the first one, if that's okay. Chair's prerogative. I have 3. I'll leave the 2 if you don't have much to say, but still. Caroline, so I come from a completely different background. It's absolutely fascinating to see all this fantastic work. And I have a very sort of pragmatic question. So it has to do with that screen between your partners. And I noticed that the screen covered a lot of the body, but not the head. So they had eye contact. And so have you tried the same experiments with the screen all the way up or down? And what are the sort of thoughts behind the sort of the height of the screen, if that's
That's
okay?
a really good question. So of course, we don't want to rule out that there are always visual cues to movement as well as auditory cues to movement. And We haven't changed the height of that screen, but we have removed the screen altogether. The reason for the screen was that non-musicians look at each other's fingers a lot. And musicians know that the fingers are going too fast to really be useful one finger movement to the next, but non-musicians don't know that. So we tried to discourage them from watching each other's fingers by allowing just the shoulders up to be visible. But the one study that I talked about today that did change it was the singers. We had the singers facing each other and singing, and then we turned them outwards so they couldn't see their partner at all. And the amount of asynchrony remained the same. Now, these are not professional singers. They are amateur singers who are comfortable singing in groups. One knows if you're singing in a group, you can't always turn and look at the person you're singing with, right? So it did not make a difference, the visual cues for the singers.
Okay. Thank you very much. Yes, we have it here first. I'll keep an eye. And on YouTube, you're also very welcome to ask questions. Just put them in the chat.
Thank you, Caroline. This was fascinating. I'm trying to formulate this as a question now. In your examples, you have been asking people to do specific tasks. And as you know, I've been interested in kind of non-tasks or kind of just everyday activities or swaying and these things. So I wonder if you could talk a little bit more about this, because also they have also found the 2 Hz resonance natural frequency or just in everyday actions as well, where you don't really do anything, you just kind of capture how people move around, etc. And could you say a little bit more about how you think that your findings here would also fit with kind of more everyday type of activities and when you're not doing specific things?
Sure. This is a really good question, and one we want to approach with bigger groups than 2. In 2, there is, if you look at Stephen Strogat's nice models of dyads versus triads, there is always a balance in a set of 2, but there's usually an imbalance in a set of 3. And so the violin study was with threesomes, where it was more open-ended, how the 3 determined how they were going to synchronize with each other. And we think we should see more of that as we get to larger groups. So I think that's the way forward for us, but perhaps you have specific suggestions, so we should talk about that. Your second question was the 2 Hz. So yes, the 2 Hz special movement rate has been seen in a lot of human tasks, but you have to take into account the limb differences. So when you ask people to do things with their legs, it's much slower than if they do things with their fingers. The fact is we are so driven by the sensors on our hands, we tend to do most tasks with our hands. And so I would argue a little bit with the specialness of the 2 Hz in that it doesn't test all types of movement. So the violinists here use a much bigger movement than we typically see with finger movement tasks. So part of that might be the task again.
Thank you. I hadn't seen the sucking reflex hypothesis before, so I'm very curious about that. I mean, it's as good as hypothesis as any. It's certainly a real thing that we have. So yeah, but I'm wondering how you're thinking about these sort of predominant fundamental rates that seem to be very present at a low level, and how that might be different between species, given that the kind of synchronization that we move to as humans is so different from the kind of synchronization we see in primates that presumably have similar behaviors and in early infancy, at least for some of these things. Where, I mean, and I don't know if anybody's done comparable comparisons for sucking rates and, you know, baby chimps, but do you have any intuitions about how that might be different across species?
Okay, so it depends what primate you're looking at. The fact is we know the most about the neural wiring of birds, and birdsong has been the most common comparison with humans in terms of auditory motor coupling. We don't know as much about other species. I would imagine, though, that given that the fact that the ones who can't suck young die, that this is a reasonable way to go. So let me give you an example that I just happen to have. Barlow did this really nice work with human infants who were in the NICU and unable to suck. And at time A and time B, they get no stimulation. They measure the sucking rate. They actually have to be fed intravenously to survive at that point. He introduces training, rhythmic training, which is an oral device to stimulate with a rhythmic pulse their sucking rate. And so he introduces it and measures it 9 days after daily training and 4 days after and 10 days after. So this is the sucking rate of the pre-training, and this is the sucking rate of the post-training at 4 days and 9 days. This is quite dramatic. So they have the ability to learn, but they need initial prompting. There are methods used with newborn like calves and sheep that use a similar pattern. They try to induce the sucking with rhythmic stimulation. And for some portion of those newborns who are not sucking, it works and carries over. That is, they can stop the training after 20 days and the infant will survive. So that lends hope to this hypothesis, but that's all I've got right now. It's a good question.
Hello, thank you. I was thinking along what is the origin of this, wide individual differences, and I was thinking all the time if there is a brain oscillator that is expressing itself in these behaviors, but then in the end you came to this sucking account. So I was wondering, why wouldn't that also be expression of something central? What do you think about that?
I think most people would argue the sucking CPG is central. It's because it's so tightly tied to, I'm going to go back here. It's so tightly tied to respiration and swallowing. And swallowing is a tough thing for a newborn to do. So you can't swallow, suck, and breathe all at the same time, right? So these oscillations have to entrain to each other. The only one, however, that is at the rate consistent with auditory motor coupling is the sucking one. And I think that has mainly to do with the bone conductance of the sound. So swallowing and respiration also have some bone conductance, not respiration too much, but swallowing does. But it's at a very low rate frequency compared with the sucking. So, and since sucking has to be in place the very first day for that infant to get nutrition, of course, respiration does too, but swallowing comes a little bit later. It's tougher. It involves more musculature too. And infants' necks take a while to develop. So that would be my hypothesis, but do you have an alternative hypothesis?
No,
Okay.
not really. I'm just considering the possibilities. So
Yeah.
thank you.
Thank you so much. That was really a great talk, and I'm impressed by the kind of the systematic approach that you have, that you kind of investigate these step by step and really pursue your ideas. That's inspiring. I had a comment because it might be that you've already, I didn't get it from your presentation, but when musicians synchronize, I think it's quite common. It's both been empirically investigated, and also a lot of musicians testified to the synchrony being more stable if there is a small asynchrony between the two sounds. It's easier to be stable if you have, like, if your own sound is not masked by the other sound, and vice versa. So I just wonder if you had, because I remember we discussed accuracy and precision after our beat pin presentation. Did you use both measures here, or is it?
This is based mainly on the mean, so accuracy, but it's also based on precision in that the standard error around the mean is small and doesn't overlap with the next person's spontaneous rate. So some of those graphs had standard error bars above the general upward trend from fastest to slowest. So it's both mean and variability that count here. But your bigger question, I think, about why it helps sometimes to be asynchronous. I have a position on this that is driven by nonlinear dynamics. And it has to do with the fact that when you have two oscillations, one is responding to the other. And if this one's oscillation is in the range of this one's, they find it easier to couple. There is a time delay that I talked about when I showed the inhibitory, excitatory, there it is, there. That feedback loop takes time. And when you look at coupling models with a time delay, it actually predicts which person will, or which of the two oscillators will fire first. So we can model the amount of asynchrony and the direction of asynchrony by changing the nature of the feedback loop. And that's what I'm going to talk about on 16th. That's a sort of long story, long answer to a big question.
I look forward to that talk.
Thank you.
Thanks.
Hi. Yeah, so I was wondering about this situation that might be familiar to many people who have been music students. When you perform music in public and you're nervous or you're highly aroused, often you tend to play faster than is necessarily appropriate for the music that you're playing. So I'm wondering whether this has been studied, the relationship between anxiety or arousal and the spontaneous production rate.
That's a really good question. So there is interesting evidence about playing faster when one is aroused. And I don't have a study that shows that, although one could argue that these non-musicians on our studies show slightly faster spontaneous rates because they are nervous. They are put in a position of having to play regularly with a partner, and they've never done this before. So I suspect that some of that physiological arousal increasing the frequency might pertain to our non-musicians. But I do have an example of decreased arousal. Okay. So in this experiment by Shannon Wright, we wanted to know if circadian rhythms dominated the spontaneous rate. So she brought in pianists to play the same pieces of music 4 times in the day, 9 o'clock, 1300, 1700, and 2100. And she measured their spontaneous rates. And you can see from the fastest to the slowest, again, in mean tone duration, upper left, we kept the same ordering from the 9 a.m. to the 1 p.m., 5 p.m., and 9 p.m. And you can see that the trend continues. There's some changes, but there's still a strong correlation between the different times of day for each individual. This is for familiar pieces. So she also had them do unfamiliar pieces. And you see the same pattern, that the pattern at 9 a.m. Tends to be the same across the day. The reason I thought of this is because they're slightly faster by the end of the day than they are at the beginning of the day. And these were college-age students who, filling out the circadian rhythms questionnaires, qualified as night owls, not early morning birds. So the night owls came in and took physiological arousal tests, and they were tired in the morning. They were awake by the end of the day. The experimenter was not awake at the end of the day, but the participants were. So this suggests that it can go both ways. It can work against you to have too little arousal in terms of perhaps suppressing a little bit the spontaneous rate.
Thank you. Thank you very much, Caroline. One question concerning temporal thresholds. You said 2 hertz and different values. One area which I am asking you could perhaps have some relation to this is a so-called psychological refractory period in terms of the updating cycle. When you speak about auditory motor coupling, that it's not instantaneous.
Right.
It takes a certain amount of time. And from the literature on the psychological refractory period that has involved this thing about intermittency in motor control, suggesting that it is discontinuous and that it's actually advantageous to control motion chunk by chunk. It's a a bit counterintuitive from what we are thought of to be the proper control model. But do you have any opinions on
I think
this?
that's a really interesting way of getting at feedback loops. I think that we should look at the time scale of the psychological refractory period, and what we've been assuming is neural transmission rate dependent. So it takes time for auditory cortex and motor cortex to signal to each other. And so the first thing we'd have to do, though, is check the time scales, because usually one would consider the neural transmission rates lower than the psychological refractory period. So I would like to talk with you about that. Yeah.
So I don't have any more. Yes, one person more on my list.
Thank you for the talk first. I have a question regarding the power spectral density plots that you show. So there was a plot where you align the central frequency, that one on the right. So I noticed these little bumps just next to the center. So I was wondering, do you think this might be related to be close to the natural frequency of an attractor? So then it's difficult to be kind of tiny bit left or right to the central frequency. Then it's not possible to have that frequency so much expressed, or it's just perhaps an artifact of the FFT windowing, or maybe something else.
It's hard to rule out those two. It's hard to distinguish between them, because we only used a methodology that did create in part or exaggerate the dips around the trough, right? So the method we used takes an average across neighboring locations. So it does end up enhancing the trough to peak difference. But the Nyquist frequency, I don't think that can account for this completely, because of the very slow rates compared to the sampling rate at which the EEG is measured. Is that what you were asking about?
No.
Not Nyquist. Okay, sorry.
No,
I
I'm
misheard.
just thinking just the windowing effect,
windowing
in the
effect, yeah.
FFT. If you change windows, perhaps that shape might change. But if it stays there, it's perhaps an attractor that is not allowing those
okay.
frequencies next to that peak.
Well, I can tell you that the shape stays the same across the left end and the right end of the top graph if we just put the window there, but that's a fairly limited range up in the top graph. So maybe I can talk with you afterwards about this. You might have ideas for how best to compare those.
Okay, thank you.
Thank you.
And a question from Laura.
Yeah, I wanted to ask about the potential relationship between the spontaneous production rate and timing when you're imagining music rather than performing something overtly. And I think there's been a number of studies that show that timing is not quite perfect when we're imagining music. And I think it usually tends to slow down, although I recall reading some studies of piano playing, and when you take the sound away, then it tends to speed up. So I suppose I'm wondering if you were asking people to imagine at a particular rate, say with one of these synchronization continuation type paradigms,
Right.
whether that would be if they would maintain the timing better if it was close to their rates.
I think we would have to predict that. Yes, that's a good next experiment. I was thinking of what you were saying made me think of Andrea Halpern's work with strong correlations between imagined rate and performed rate, but that's a correlation. So the intercept is a thing of issue here. Are they truly imagining faster? And most tasks do come out imagining faster, but you could argue that's the motor system time to make those movements. So, but you're right, we would probably have to argue that the spontaneous rate should hold in imagined tasks, that it should at least be easier to imagine at the spontaneous rate than at faster or slower rates. Whether or not I'm not sure what the dependent variable would be here, because we would want to measure temporal stability, and it's hard to do that in an imagining task without a motor response. But maybe you have ideas. So I have lots of conversations to have after this talk.
And it's great. We have a question here from our online world, Celisti83. So the question is, sorry if I missed it, but did you look at the potential contribution of body size, height to spontaneous production rates?
Yes, we did. I'm glad you asked that question. So body frame, as measured at the ulna, is supposed to be a good indicator of some findings in temporal coordination and motor tasks. And we found some evidence that the ulna width was correlated with the spontaneous movement rate in the absence of sound, but not with the spontaneous production rate in the presence of sound. This is work by Becca Schurig. So that suggests that body frame is more related to straight movement characteristics, not necessarily those that are sound producing.
Okay, I think we are moving towards our reception where we'll have all those conversations that we've now promised. I have a couple of questions further as well, but would everyone just like to join me in thanking Caroline for a wonderful, wonderful paper?